This is Gene Veno from the Susquehanna River Basin Commission, and hopefully you tuned into our first segment on the Billmeyer Quarry in Lancaster, Pennsylvania. And today we'll be launching part 2 of this program. As you can see, some of the slides that we are putting up. When we left off last week, the project manager, Pierre, we had learned about an existence of an invasive species, and I wanted to spend a lot more time on this subject, and that's why we're into part 2 of the Billmeyer Quarry project. So with me today, again, I have joining us is Miss Ellyn Campbell and Miss Brianna Hutchinson from the SRBC Monitoring and Assessment Program, a very busy program that on a day-to-day basis could have 50% of their employees in the field doing such programs as we're talking about. So Ellyn and Brianna from SRBC, why don't you introduce yourselves again for our viewers who weren't with us on the first series. Ellyn?
I'm Ellyn Campbell. I'm the supervisor of the Monitoring and Assessment Program at SRBC, and I oversee several of our aquatic biologists.
Brianna?
Hi, yes, I'm Brianna Hutchinson, and I am one of the aquatic biologists in Ellyn's program, and I was in charge of the data collection for and monitoring for this part of our project.
Well, thank you both for being here today. You were in part 1 of the program, but there wasn't much to talk about until we got to that one question about this invasive species that showed up during the beginning of the project. And so I said to Pierre, I need to know more from Ellyn and Brianna. So what exactly did you mean by invasive species, Ellyn and Brianna? Would you like to start off the program, and what are we looking at here for our viewers?
Well, so right on the screen right here is one of the invasive species that was found at the quarry itself. And to back up, an invasive species is a species that isn't native to a particular region. And it's called an invasive species because its introduction to an environment causes harm when it's introduced to the new area.
Well, I recall correctly from our previous part 1 interview with Pierre that it was a species of mussel, something to the nature that you found in the quarry. So you and Brianna provided these photos, which I appreciate for today's program. But let's pull up something else. You have what is a quagga mussel? I'd like to have you speak to that. Are they harmless? I mean, what can you tell me about these mussels?
So quagga mussels are related to zebra mussels, which are something that maybe people are more familiar with. They're a species of freshwater mussel that's native to parts of Eastern Europe. Zebra and quagga mussels came over on boats and were first introduced to the Great Lakes area, and they've spread since then. And they're filter feeders that attach to stationary surfaces like rocks and aquatic vegetation. And you can see from the picture that they're actually really small. They're only about 1.5 to 2 inches long at maximum. And they can cause big problems when large numbers of them attach to boat docks or water intake pipes or other structures that are located within the water. And unfortunately, then that makes for a lot of headaches for fishermen and drinking water suppliers. And there is a lot of money that's spent trying to troubleshoot getting rid of them or controlling them. And they also reproduce really quickly. That's one of the other factors that makes them an invasive, a very effective and productive invasive species. A single female can produce over 1 million eggs in a single breeding season. And because their populations can expand so quickly, they can outcompete native mussels that might be present. And many of the mussels are already endangered here in Pennsylvania. So it's a big problem. Problems range from economic, like I mentioned earlier, to environmental.
I understand economically in the millions. I also, like you said, Eastern Europe. Is it Ukraine? Is it where it started, ended in Utah? Right. And so I guess the question, how do they get in Lancaster, Pennsylvania?
Well, so one of the things to note about zebra and quagga mussels is that a lot of times you'll find them in inland waters, like in Pennsylvania, at places that used to function as scuba diving schools. And because they're amazing filter feeders, quaggas are miraculously introduced to these areas to clear up the water, which makes them more advantageous and desirable for the scuba divers. And prior to the Lancaster County Solid Waste Authority acquiring the the quarry was used as a scuba diving school. So who knows, they could have been accidentally introduced by boaters, or they might have been put there on purpose. We don't really know, but all we know is that they're there, and we couldn't really go forward with the project until we figured out a way to get rid of them.
How is that such a concern? I mean, you mentioned about what they do and the millions that they cost all across the nation and worldwide in eradicating them, but how did you get started? Like, couldn't you just, not worry about them?
Well, not really. Now, because they were in the quarry, they're situated in this isolated body of water, and that would have been fine the way it is, except that what we needed to do was to test the amount of water in the quarry by doing this pumping test that Pierre talked about. And that would be fine, except that quaggas have a free-floating life stage called a veliger. And I think we have a picture. There we go. They're teeny tiny. They're so small. They're only about the width of a piece of paper, and you actually need a microscope to see them. And SRBC didn't want to take the chance of pumping the water from the quarry into the river that contained veligers, because there are currently no records of quagga mussels in the river downstream of the quarry. And by us introducing veligers in the river, we might jumpstart a population, and we didn't want to have that happen. So it was important to do everything we could to keep them out of the river.
I mean, that's a major environmental undertaking, and the SRBC, you, Brianna, and Pierre, to be really complimented for looking not only just for the project, but for the environment and the people that depend on the Susquehanna River. They're microscopic, as you said, so how do you go about removing them?
Well, it's not so easy, and a lot of times eradication procedures, they can be expensive. So we partnered with Earth Science Labs and Solitude Lake Management to eradicate the mussels by using a copper solution that was applied to the entire quarry. And this solution was specifically targets mussels and isn't harmful to any of the other species in the quarry, like all the fish species that are there. And Solitude treated the quarry with the solution 3 times during the summer of 2017 before the pumping test took place. And I think we have a picture.
Yes, I think we do. The next sentence.
There we go.
So now what we are saying here, you had to do a quagga mussel treatment, and then take a pumping test, and then place almost, going back 3 years to 2017, the quarry had completely refilled since then, right? Is that correct?
Yeah, but before that, I just wanted to point out the picture of the application there. You can see the blue copper fluid that's in there.
Okay.
Yeah, and when that was pumped into the quarry itself, that it was very successful. Within a few days of the first application, we started to see dead mussels floating to the surface. Yay. So we were very happy about that, because if you kill the adult mussels, then that chances are that means that you killed the veligers as well. And so we had Solitude and some contractors took some plankton samples. So they basically ran a net through the water to collect the microscopic life and to look for the live veligers, but they only found dead ones, which is exactly what we wanted to see. And it was at that point that we felt confident that we could go forward with the pumping test and put the water in the river and not have to worry about any downstream populations establishing themselves.
So those containers were filled with that treatment.
Right.
And I've been to the quarry, which we're going to get to a picture. And are you saying that whole quarry was treated with this element?
Yes, they applied it 3 separate times through, I think it was like a 30-day period, more or less, throughout the entire surface. They had a grid pattern that they followed. And what's really cool is if you go to the next slide, you can see that after the pumping test was done, which, as Pierre talked about, removed I think it was 60 feet of the upper part of the quarry, you can see what the extensiveness of the population of the quagga mussels was, because now all the areas where the mussels were living were now exposed. So you can see them clinging to, I don't actually know what that piece of structure there is, but you can see them all dead and gaping. And so it was kind of a 1-2 punch to make sure that we got rid of the quaggas. And if we had gone ahead without first eradicating the mussels, like I said, then it would have been a big problem for the river.
The picture behind me again is a massive, massive site. I've been there. I've actually been on one of the boats that Brianna captained, and I got to see firsthand what she does. And I'd like to bring Brianna into the discussion here if you can. What is your role there, Brianna, at the site now? Once we have the quagga mussels eradicated, we have the pumping tests are done, things are looking good. Now you come into play. What do you have to do?
Yeah, so as I mentioned earlier, I've been the manager of field monitoring operations. So last summer, we established a monitoring program to sample for the presence of quagga mussel DNA in the quarry. So the technical term for DNA that is just detected in the ambient environment, such as the water in the quarry, is environmental DNA, or eDNA for short. And eDNA can pretty much be any biological material that's shed by an organism into its environment. So in the case of quagga mussels, the eDNA could be cells from living or even dead mussels, or it could be waste products from live mussels and veligers that are then shed into the water column.
Wow, the three of you, I think our next slide will show something here of, so you're actually in a boat, you have me there, but what you're doing the eDNA sample collection. I think I'm dragging something on that pictured slide there. What am I actually doing?
Yeah, so starting last summer in July, my team and I went out to the quarry every other week through the end of October, and we were collecting these eDNA samples. We ended up making 8 trips in total. And these pictures are just showing the actual eDNA sampling process itself. So it's pretty straightforward. We have just one person who's holding a net that's attached to a long piece of rebar into the water, while another person is piloting the boat. So the picture of you and I, we're piloting the boat. And then as you can see in the photos there on the right and the bottom left, the net is towed through the water alongside the boat. So the net itself has a very fine mesh, and there's a cup that's attached to the end of that net. So all of the materials that are in the water column are being funneled through that fine mesh net into the collection cup. So that could include stuff like plankton, algae, and even microscopic fragments of the environmental DNA, which is what we were sampling for.
So now that you've collected the sample, you're processing, it's involved to a lab, is that correct?
Yeah, I was just gonna say, thanks for the next slide there. So after we collect the sample, we then have to use water to rinse the net and get anything that might be stuck to the sides of the net down into the cup. Because as I said, it's a very fine mesh, so it can get clogged easily. So we have to make sure that we rinse that really well, get everything down into that collection cup. So that's the picture there on the left. Jean, you might remember that. You were helping me with rinsing the net. So that's
and you were very specific, like we had to wear gloves, you had to make sure it didn't touch anything. Is that all correct?
Yeah, so we wear gloves whenever we're processing the samples to prevent cross-contamination between the samples. So we actually, if you remember from the bathymetric survey picture, there was 2 kind of distinct areas of the quarry. So we were collecting one sample from each area of the quarry. And we have to wear gloves, again, to make sure that, we're not contaminating between samples. So once we have rinsed the net itself, then we need to rinse the contents of the little collection cup into our actual sample bottle, which is just a small plastic bottle. That's me in the photo there on the right, rinsing the sample into the bottle that we would then send to the lab. So we use ethanol for that final step, because that ethanol will preserve any of the DNA fragments that might be present in the sample without degrading them, so that they can later be looked at by the laboratory professionals and see if there's any quagga mussel DNA present in those samples.
So Ms. Campbell, Ellyn, she determined there were these invasive species, and now we get to that point. Now you're actually, after they have been eradicated, we're now doing the testing. So I have a question for Ellyn and Pierre. Was there some intensity of worrying? Were they all eradicated? Did you have that concern, Ellyn, at one point? And was there some good news that came from all of this?
Yeah, you could say there was a lot of worrying that whether or not they came back or not, because the success of the project and the success of being able to use the water basically hinged on us being able to say that all the quaggas were gone. So we did get results back from the sampling that was done last summer, and through all of the samples that were done, we did not find any environmental DNA of quagga mussels. So we're going, yay!
And that's when was that part of the sampling that we did that day, Brianna? Was that part of that? Was that the testing that you did?
Yeah, I think we took you out in September. So
yes.
That was about halfway through our sampling period. It ran from July through October.
Wow. So, Brianna, before we conclude segment 2 of this fascinating story that many people, I think, will be just delighted to see what you did with an abandoned quarry. It sat idle for many years thereafter with water just filling with quagga mussels. You eradicate the quagga mussels. You're doing the testing. You're getting great news. What does the future hold for your sampling procedures, and what do you have to do, Brianna?
Yeah, so we have to continue to make sure that there's no evidence of quagga mussels in the quarry. So we do plan to repeat that sampling that we did last summer. We plan to go back out again this summer and also in 2021. And we'll be keeping the same schedule, so the July through October timeframe, bi-weekly sampling. And we've also talked about doing some additional water quality monitoring to see if there's any changes occurring in the water chemistry within the quarry now that the quagga mussels are gone. So that's will hopefully be an additional aspect that we'll be looking at. Because like Ellyn said earlier, the quagga mussels, they're such good filter feeders. They can have a lot of impacts on other things within the environment, such as like the turbidity of the water, how clear it is, and things like nutrient cycling. So yeah, these are all things that we plan to continue doing. Make sure that there's no quaggas, they're definitely gone, so that we have this water resource to use in times of drought in the future.
Well, Ellyn and Brianna, Pierre, I mean, this is a project that needs to be viewed by many people. The success story, the environmental success, the protection of the environment. Once you determine that there was invasive species, these little quagga mussels that cause millions of dollars. I mean, the research on these have created all across the country when we read about the boats and the marinas that have suffered. I give you really a lot of great compliments, all three of you, for taking on this massive task. And I want to conclude this part of the program, Series 2, on the program that got us to the step of having this water storage unit in Lancaster County. Behind me again is the picture. I look forward in our next segment to give a flyover that Pierre was able to commission a drone to fly over the quarry. Once you see the massiveness of this quarry behind me, I think you all too will see what I've seen firsthand, a true success story for the Susquehanna River Basin Commission. This has been Gene Veno at SRBC, along with Pierre MaCoy, Brianna Hutchinson, and Ellyn Campbell. Thank you for participating today.